I'm here with Patrick Reardon, who is the third Wiley postdoctoral fellow. Patrick's from Duke University. Welcome to EMSL, and why don't you tell me a little bit about yourself?
All right, so I started my undergraduate career at Oregon State University, which is in Corvallis, Oregon. I grew up in a suburb outside of Portland, and then I went on after working for a couple years as a technician at Oregon State. I then went on to do my PhD work at Duke University in the Department of Biochemistry. There I used NMR spectroscopy to study a number of different biological problems, including structure and function of proteins in living cells, as well as structure and function of proteins on membranes.
Great. So what attracted you to the Wiley Fellowship and to EMSL in general?
Well, I thought that EMSL really had a unique set of equipment and expertise that I thought could be very valuable for furthering my own career, as well as providing new techniques to study protein structure and function on membranes. We're here in the NMR Center at EMSL, which was a big draw for me, where we have a lot of state-of-the-art high-field NMR instruments that we can use to study proteins and protein structure. I was interested in learning solid-state NMR spectroscopy, which is a big capability here at EMSL, and so I thought that was well worth pursuing. I also like the extensive microscopy techniques that are available at EMSL, things like electron microscopy and fluorescence microscopy, which can also be a vital part of understanding protein structure and function.
Yeah. Cool. So what are you working on? What kind of projects are you working on now?
So right now I'm actually working on a project that I started at Duke with my advisor, Len Spicer. And this project has to do with the structure and function of a membrane-associated protein from HIV-1. First off, I just want to make it clear that we're not actually working with live virus here. That's not something I'm particularly interested in doing at this time.
Me neither.
Yeah, right. So what I'm actually doing is the region of HIV-1 is that I'm interested in is on the surface of the virus particle. And this is a target for broadly neutralizing antibodies. And it turns out that this particular region exists in a trimer. And so part of my project was actually designing a trimeric form of this protein that was small and truncated enough that we could use it not only to study the structure and function of this protein, but also as a potential vaccine candidate. And it turns out that this is actually in vaccine trials. It's been in small rodents and whatnot. And so we're continuing that collaboration with the folks at Duke.
So I'm a little familiar in talking about this with Len, and so this is the part of the Gates funding that he's got. And so the ultimate goal is to make affordable vaccines for folks in third world countries. And
yes, that's right.
And that's a great thing to be working on.
And,
so
we're hoping that we'll have some success with this.
Yeah, that's fantastic. Great. So you mentioned Len, who's a longtime friend of EMSL, chair of our science advisory committee. So you probably had a pretty good idea of what EMSL was like. What was your biggest surprise once you got here? You mentioned the microscopy. So was it the ability to bring all of these? Did you really think that you could bring things together?
So Len had alluded that one might be able to do that here, and I guess I was a little skeptical at first, but I think that having gotten to know people around here at EMSL, that it looks like that's going to be quite possible to do.
Yeah.
And so especially during my interview process when I talked to like Baron Cavius and whatnot of the microscopy group, it became clear that I think that people here are interested in that kind of collaborative research, and I think that's really helpful.
Great. When I do these interviews, I always let the interviewee ask a question or two. You got one for me?
So one of the more exciting things that's just happened recently is we've been working with Bruker, the NMR vendor, on dynamic nuclear polarization. What are the chances of getting a DNP instrument here? I
think that's something we definitely want to explore getting. And so by doing the experiments with Bruker that you guys are doing now and testing out the types of analyses that you can do, it's going to help us build our case for BER, which is ultimately what we have to do to get the capital money. So what I can do is tell you I'm committed to trying.
Great.
And that's what we want to do. I think it would be a really nice fit for to complement the capabilities that we have in here between the the NMR and the EPR, and so I'm committed to trying. But So you got to help me build a good story.
All right, we'll do that.
All right.